Hello, and welcome everybody to Gene Editing and Myotonic Dystrophy. This session is part of the DM Professionals track. It is intended for clinicians, scientists, and industry experts. This session is being recorded and will be available for viewing after the conference on the MDF Digital Academy at www.myotonic.org/digital-academy. I'm Dr. Nadine Skinner, the MDF coordinator, and I'm delighted to introduce our panel chair, Dr. Ashizawa, today. To ask our presenters questions during the presentation or to make a comment, please use the chat function. Dr. Ashizawa is our panel chair and the director of neuroscience research at the Houston Methodist Neurological Institute. He graduated from Kiowa University in Medicine in Tokyo in 1973. He completed his internship in medicine in Pittsburgh, his residency in neurology and fellowship in muscular dystrophy at Baylor College of Medicine in Houston. While at Baylor, he assisted one of several teams around the world in locating the DMPK gene for myotonic dystrophy. He's been given numerous research awards and grants, especially for myotonic dystrophy and ataxia. In 1997, he and Dr. Claudine Junian co-founded the International Myotonic Dystrophy Consortium, IDMC, a biennial scientific meeting where physicians and scientists convene to determine the cause and ultimately a viable treatment or cure for myotonic dystrophy. Dr. Ashizawa has published numerous articles on myotonic dystrophy in medical and scientific journals, and we're very excited to have him chair this esteemed panel on gene editing and myotonic dystrophy. Dr. Ashizawa, welcome.
Thank you very much for such a kind introduction, and it is really a great pleasure to be here and chair this session. And so this MDF conference is getting better and better in spite of this world pandemic of coronavirus. And indeed, I was talking with the speakers prior to this session started, that we can review all the talks with no pain, and then we can join the discussion. And even after the meeting, we can do that. I mean, this is really empowering the intellectual interactions, and I'm very excited about it. So we have two speakers today about gene editing. And so first, I think Vincent, you go first, right?
Yeah, sure.
So first, let me just introduce Vincent Dion, Dr. Dion, and he is I personally knew him because he was in Houston. He got a PhD from Baylor College of Medicine, and he was mentored by Dr. Zhang Wilson. He was a professor at the Baylor College of Medicine. He still is, but he retired. And he plays golf a lot. And tomorrow I'm playing golf with him. So I will report the performance of Dr. Dion to Dr. Wilson. But anyway, so we all have a sort of a Houston connection. So his work on the mechanism of a CAG-CTG repeat expansion is quite well regarded. As a postdoc with Dr. Susan Gasser at the Friedrichs Miescher Institute in Switzerland, he worked on the role of chromatin remodeling in DNA repair. He started his lab at the Center for Integrative Genomics of the University of Lausanne, which is Switzerland again, in 2013. And moved to the UK Dementia Research Institute at Cardiff University in 2019. As you know, Cardiff is deeply connected with myotonic dystrophy. It contributed to a gene finding, gene discovery in very heavy fashion. And the late Professor Peter Harper was a giant in this field. And so Dr. Dion's lab is making key contributions towards the development of gene editing approaches for expanded repeat disorders, including myotonic dystrophy. Okay, Vincent, go ahead.
Thank you very much. So the pressure is very high at this point, given this introduction. And can you see this properly? I think so. Okay. So, yeah, so, and the other thing I'll do is I'll start by actually citing a paper from Cardiff back in 1993. This was not planned, but it's been known for a long time. This is the plot of the age of onset plotted against the size of the repeat track. And I'm going to try to move my wires out so I can see. And so what you can see is that if you have, if individuals have a short repeat track at the DMPK locus, the age of onset is relatively late. And as that repeat size increases, you end up with a very early age of onset. And so this is what we heard from Darren Monkton and Christopher Pearson yesterday. And so what I'm more interested in is this idea that you can, if the longer the repeat track is, the more severe the disease, then if you were to remove it altogether, you would reduce the severity of the disease. Here you would remove the underlying cause of the disease. And so if you think about this, you could be inducing contractions, or you could be excising it out altogether. You want to make sure that you don't have expansions that you're causing at the same time, because then that would nullify any benefits you would have from the contractions or the excision. You're trying to avoid mutating the non-pathogenic allele as much as possible, because those are likely to have some physiological function. And you want to try to avoid off-target mutations. We are talking about a DNA damage inducing agent, and so you want to make sure you control it as best you can. And so, and then finally, you want to try to have it effective in as many individuals as possible. And so with that in mind, there's a number of groups who have excised the repeat track successfully. So in a variety of loci and a variety of different systems. And I'm going to say that this cartoon is a gross underrepresentation of the complexity and the sophistication of the studies that have been done. But I think it illustrates what is being tried here, which is trying to cut with an endonuclease called Cas9 on both sides of the repeat. And you're hoping for an error-prone repair of that removes this region, including some flanking sequences. And so for this to, so the good news, I should start with that, is that the first, sorry, I'm getting sidetracked by the chat. I'll focus here. So the first thing is that it works. So if you remove the repeat track and you look for example, splicing defects, so those tend to be corrected or at least ameliorated in cells. And so this is the really good news about this. The downside is that here we have two different guide RNAs, and therefore twice the potential of targets. If you want to make this allele specific, you have to target SNPs between the chromosomes that may or may not be present. And it's not terribly effective overall. And so what we ended up doing was to take a different approach and to try to nick the repeat track itself. So we have a guide RNA that targets the repeat, and then we're introducing nicks rather than double-strand breaks. Those are much less mutagenic, and this, we hope, leads to an error-prone repair that shrinks the repeat track, but nothing else. And this is that approach that I want to talk about. When we first started this, we took from John Wilson's lab a reporter gene that contained 2 GFP exons and an intron in the middle. Inside that intron, there's a CAG repeat, and this whole cassette is a single integrant in HEC-derived cells. So this is not a model for the disease, it's a model for repeat instability. And the way it works is that if you have an expansion, this repeat interferes with the proper splicing of the reporter, and that leads to cells that are not very green at all. By contrast, if you have a contraction, that restores the splicing and you end up with green cells. And so we can use this very easily to screen for treatments that would induce contractions. And so, for instance, the way we do this, just to give you model data so that the bar graphs are actually quite more complicated than they look. And so what we do is we take a culture of cells, we transfect in Cas9 plus the guide, and or in this case, a control without a guide. And then you end up with a distribution of GFP expressions. And you can draw gates that correspond to the dimmest 1% of the cells or the brightest 1% of the cells. If you sort those, you get expansions in the GFP minus gate, and the GFP plus gate contains contractions. And so what you can do then is do in parallel. Oh, and then this is all the data is normalized to this control experiment at 1%. Then what you do is you do the experiment itself with the guide RNA against a repeat, and then you look at how much of your new distribution falls into the GFP minus or the GFP plus cells. And you can do this multiple times, and this is how you get those bar graphs. Okay. So if you use a Cas9 nuclease that induces a double strand break within the repeat track, what you see is that you get small increases over here and over here in both the GFP minus, so the expansions, and the GFP plus, and those are the contractions. So this is not super convincing or desirable in any way, but when we did the same experiment with the Cas9 nickase, so the same guide RNA, but now we have a point mutation in the Cas9 enzyme that turns it into a nicking enzyme rather than an endonuclease. What you end up with is a bias towards contraction. So you can see here with the two guide RNAs against one side, one strand of the repeat or the other, you get robust contractions. You can move away from the reporter gene and look at the DNA itself using the gold standard, which is a small pool PCR. And this is what Darren was showing yesterday, so I won't go through how we do this, other than to say that here we have a cell line that's been transfected 3 times with the Cas9 nickase together with an empty vector. And so what we're looking at is background instability. And so that is what it looks like. And then if you do the same thing with a guide RNA against a repeat, you can see that you have a lot more contractions down to repeat sizes that are in the non-pathogenic range. So this is nice, but we wanted to know whether it was A, locus specific, and B, cell type specific. So we got some lymphoblastoid lines from DM1 individuals and then infected them with the Cas9 nickase and then with a guide RNA, and then took time points. And so what you see here is that this individual has 2 alleles, one with 1,600 repeats on one and one with 5 on the other. And what happened is that within 5 days, we had lost the expanded allele. And by day 30, we had very few expansions left. I have to say that this is surprisingly efficient, and not all cell types do this well, but it illustrates the fact that it works at multiple cell types and in multiple different loci. And so this is actually quite a bit of good news. Then we went back to the reporter assay and we asked the effect of the repeat size. So this is important again because patients are heterozygous, and so you want to try to keep the non-pathogenic allele intact. And so what you see here is that as you increase the number of repeats, only the ones that are very large, at 100 and 270 repeats, actually contract properly using the Cas9 nickase, making this inherently allele specific. And so what we think is going on, so I'm not going to go too much into the mechanism of how this works today. I'm just going to have this slice to summarize it. What we think is going on is that if we have single nicks within a repeat tract that's in the non-pathogenic range, you end up with simply an error-free repair of that. On the other hand, when you have an expanded allele, you can end up creating multiple different nicks along the same strand of the DNA that can be turned into a DNA gap. From there, this DNA gap can fold into a secondary structure and you can get ligation on the other side, leading to a heteroduplex. And that heteroduplex can be processed further with endonucleases or replicated and to lead to a contraction. And so I want to rather spend my time on one mechanistic observation that led us to do all sorts of other things downstream that were quite interesting and useful. So especially in the context of the safety of the method. So here we went back to the reporter, and as it turns out, the reporter has an inducible promoter. And so we can test whether transcription is required for the contractions or not. And so here what you have is the flow of the experiment, where we can do a first transfection on the first day and then culture the cells with or without doxycycline to induce transcription through the repeat track until Cas9 is completely gone in the culture at this point here. And then you turn the transcription back on to be able to read the expression 3 days later at steady state. And so the results that you see here, essentially this last bar here is the only one above background. And this is where you have the transcription through the repeat track and the Cas9 nickase being expressed and the guide RNA. In other words, the conclusion from this is that 100% of the contractions need are dependent on transcription. Okay, fair enough. So this was really really fun, because what it meant is that endogenous repeats should not be targeted. So what that means is because they're too short, and this expanded allele on the reporter gene should be stable until we turn on the transcription through it. So what we did was to make stable cell lines that express the Cas9 nickase, the guide RNA, and have the reporter, then add doxycycline to turn on transcription and watch the repeats shrink. And this is what the experiment is here. So those are just regular PCR, so they exaggerate a little bit the contractions. So just keep that in mind. And what you see is that we have 3 lines, one with 100 repeats, but it has no guide RNA. One has 12 repeats in a guide RNA, whether the transcriptions is off or on, not much is happening. And then the last one here is where the action is, where you have 120 repeats to start with. This is the top end. And as you, after 1 week, you can still see the contracted. Then after 2 weeks, you get a smear, and that's even more pronounced with an accumulation here at the end. And so this was exciting when we first got that. And it helps us actually looking at safety. And by safety, what I mean is 2 things. One is off-target mutations and rearrangements at the locus where we're getting contractions. And so starting with this latter one, what we asked was whether there were large-scale rearrangements at the repeat track upon nickase-induced contractions. This is motivated by a number of studies. One striking one in yeast looks like this, where what you're looking at is a southern blot of different colonies of yeast looking at a locus where Cas9 had induced a double-strand break. And what you see is you get lots of very large deletions, rearrangements, often that go outside of the region that they're looking at. This is not unique to yeast. We've seen this in mammalian systems as well, in human cells, with inducing double-strand breaks. And so an important question was whether we were getting this with the nickase. So for this, before I can give you an answer, I have to often open a parenthesis about bioinformatics, because what we ended up doing was to go to sequencing to try to understand how these rearrangements might be happening, or whether they were there or not. And so we ended up going with trying to overcome a major obstacle in the field, which is counting repeats and in a heterogeneous population of molecules. And so we came up with an algorithm that's called a repeat detector. And it, instead of doing what a lot of the other algorithms are doing right now, which is have a library of reference sequences that you align each of your reads to one by one until you get the best match. Here we're using a circular profile. And what that means is that you can use the information from your previous alignment to the next one. And you end up for each read scores that match the size of your repeat track. And the one with the highest score is the one that is correct. And this works fairly well. We've now tested it on our GFP assay, as you can see here. We've tested it with Huntington disease samples that we've sequenced with PacBio long read sequencing, and from MySeq from Darren Monkton's method with samples from Leslie Jones in Cardiff. And so, and what you see is that we're essentially reproducing what's been seen in these samples before. The advantage that we have, though, is that we can go to multiple different loci. We're not bound to the HD or the GFP locus. And so then what you do is you take that and you insert this information of repeat sizes into structural variant calling. So rearrangements will be essentially the same as structural variants. And so this is what you're trying to figure out, whether there's now a structural variant after nickase-induced contractions. The problem is the presence of the repeats. Because they're heterogeneous, you end up with these algorithms picking up these changes in repeat size rather than picking up actual rearrangements that we're looking for. By inserting repeat detector, in the pipeline, what we've done is essentially take out that variation in repeat size, and we can separate alleles, for instance, based on sizes that we've identified. So this worked very well. We can do things like looking for interruptions in myotonic dystrophy individuals. So this is from Stéphanie Thomé, who has a really fun poster about this with PacBio sequencing. She had data and samples where they knew they had interruptions, so we could use this to see whether we could pick them up with our repeat detector algorithm. And we do. So this was encouraging. It meant like we could see changes, and so then we could go back and ask the questions of whether large-scale rearrangements at repeat track exist upon nickase-induced contractions. We use these very samples and to look at whether we could see this. And so this is what it looks like when you plot the number, the read count, which for PacBio is called a consensus sequence, or CCS, on one axis, and on the other, the repeat sizes. And so you can see in blue is before the experiment. This is our starting population. In green is what happens if you don't have transcription, and in red is what happens if you have the transcription that's on. I'm just going to point out that this peak here is at 6 repeats for anybody who's interested. Okay, so what did we find? We ran this data on our structural variant calling pipeline, and we found absolutely nothing. There is no rearrangements in this data set. It's not perfect. It's a PCR-based library preparation, but as far as we can tell, all we get is clean contractions of the repeat track. Okay, the second part of the safety issue is whether the nickase induces off-target mutations. So this we went to, and this is done. We did 2 things. One is to start with, at the time where we described the method, we isolated clones, 9 different clones that had a contraction. So we knew the nickase was active, and then sequenced 7 endogenous loci with short repeats. So that's at least 126 loci, and alleles, sorry. And then we found no changes in repeat size, no changes in repeat composition, and no mutation in the flanking sequences. So what that suggested was that the off-target mutations were very low. They were below the detection level for this assay. So then, but 1% detection level, the 1% mutation rate is actually quite high. We don't know how far below we are from this. So then this is data that I'm about to show you that we got yesterday evening. So bear with me. This may very well change. But I'm excited about them. So I will just tease you with them. So what we did, and this is in collaboration with the Premier Alam in Quebec City, where what they did was to culture primary DM1 myoblasts. So we have run this analysis in 2 different lines right now. There's more coming, but this is all we had time to analyze for today. And so we cultured them for 1 month with or without both the nickase and the guide RNA. And then we did whole genome sequencing. And then we did 2 different things. One is we sorted out all the loci with at least 6 CTG repeats, which we evaluate to be 137 loci that could potentially be nicked by the Cas9 nickase. And then we ran the structural variant calling pipeline I just showed you on these loci. The other thing we've done was to look at the whole genome and do SNP calling with 2 different SNP callers. And that's the same thing we did with the variant calling, is to make sure that what we see is real. If it's being called by 2 algorithms, we call it real. If it's not, then we call it an artifact or something to look into some more, at least. So that gives us the structural variants. We are at any one locus where our limit of detection is about what is this? We'll say 0.008 of allele frequency, right? So 0.08%, I guess. And for the mutations, we are at 5%, but we're 5% at any one locus, right? So and then here we're looking at the full genome. And so what did we find? We found no mutation whatsoever anywhere. The reason why this is preliminary is that we still need to check for the nickase expression, for the guide expression, and for the size of the repeat tracks at the end of the experiments. So there's the possibility that we have, right as I speak, that may or may not be the case in a couple of weeks, is that we may not have we may not have the nickase expressing right now, right? We don't know this. If it's not expressing, then it's normal that we don't find any mutations. If it is expressing, it's very good news. So this is where we are. It cannot be more preliminary than this. So I think what I've shown you is that this Cas9 nickase can induce contractions while avoiding expansions. We don't touch the non-pathogenic alleles, and that is related to the mechanism by which we don't find off-target mutations, at least in one assay is solid, and the second needs confirming. And because it's not locus specific, and because it doesn't rely on SNPs between chromosomes, this is applicable to a large number of people. And so with that, I just want to leave you with some of the outstanding questions that I was hoping to be able to answer, at least the first one today, but that's going to be for another day, which is whether it occurs in non-dividing cells, right? So, or in vivo, because that's really where we want to go forward with this. The other aspect is the molecular mechanism of contractions that are going to be important. And that's not just for curiosity, but for 2 different reasons. One is we might be able to modulate the efficacy of the contractions. For example, an inhibitor of an inhibitor would work. And on the other hand, it could also be that we'll be able to stratify patients based on their genetic background in a way that we can sort out through people that would be most benefit from this and would least benefit from it. And with that, I would like to thank the people that did the work. There's lots of collaborators that are working on this with me. I have to thank the people that have done the work. So specifically, all the beginning with the transcriptions was done by these 3 marvelous people who are no longer with me. And then Alvaro has been funded by the Myotonic Dystrophy Foundation to do some of this work and in cultured cells and in vivo. Ali has been doing all the bioinformatics with some help from Eleanor, and Emma has been helping with the sequencing library preparations. And so with this, I would like to thank all of you for listening to me, which I hope you did, but I can't tell. And I'll take any questions. Thank you.
Okay, very interesting and promising results, and congratulations for the success. So there is from Alok Kumar Behera. Is it possible to use repeat detector for other RNA expansion diseases? As we
Yes.
say, yeah, okay.
Yeah. I mean, we've tried it on ALS, the C9orf, for instance. So that works just fine. I should mention that. So there's no real reason. It works on any repeated locus. So if you're interested in telomeres, that would work there as well.
Okay, very good. So another question is DM2. The person asking question is DM2 carrier. Thank you for all groundbreaking work you do, and I look forward to medical applications for DM2. That's a golden question you asked, and so someday soon it will come. He hopes, and imagine a virus helping people for change. And so what do you say about the actual application at the bedside? How far are you, and what's the delivery system and other things?
Yeah, I've stopped promising a timeline because I've realized how much things other than the science that needs to happen as well. Coming from a very fundamental background, this has been a revelation for me. It's amazing how anybody can manage to get a clinical trial up and running. So I think the next steps. So we have to, if it works, right? Because it needs to work in vivo. If it works in vivo, the next step is to start doing things like optimizing delivery, how you deliver it, where you deliver it, what kind of, and all the minutiae of the delivery vectors or systems. Then you go in large animals, and then you heard yesterday what it takes for people to bring things towards the clinic. So all of this toxicity studies and all of the rest of it would have to be done on a scale that goes much beyond my own lab. And so so it's a complicated issue.
So I understand that you had received the MDA grant and to do this, and that was translation. So is there any effort to put together sort of a team of investigators from very basic like yourself to animal studies to preclinical study to sort of acquisition of IND from FDA or EMA or something like that? Any effort going on, concerted effort?
So there's a concerted effort. There's a group of us, I should say, so that I've shown on my last slide, which is gone now, but that there are people in Quebec City, people in Paris, for instance, and us. We are in mice at the moment to try to test some of these. We're going to be trying in, and so some of that work has been funded by the MDF. The safety, the last bit that I've shown you was funded by the MDF as well. And so the mouse work is not as far as I would like it to be. We've run into every problem people run into delivering Cas9 in vivo on an AAV vector. And so, but slowly we're making progress with this, and then we'll be able to know. But yeah, so there's like, I think we're at 4 or 5 labs now helping out with this. So yeah, so I'm not by myself in my corner. I guess is what I want to point out.
Okay, couple more questions. One's from Matthew Disney. And so Matt said, do you think enough of these systems will get into muscle to affect disease? Biogen Ionis ASO was smaller and wasn't able to achieve high enough dose, and suggests that this could be a challenge. What do you say about that?
Yeah, so the muscles are difficult. That there's no question, in the sense that for AAV delivery in particular, there is just a paper out looking at this, looking at best capsids to do this, like just new AAV capsids to target the muscle specifically. So we're going to be down to, you know, delivery methods and really trying to optimize this. And this is really where my own expertise falls apart. And so if you have if you have expertise with this, I'm happy to talk to you. Yeah.
Okay. Another question is from Tom Cooper and said, what about your thoughts on efficiency in non-dividing cells?
Yeah, no, so this is ongoing. So we think it happens, but I'm not yet confident enough. So along with the myoblasts that I just showed you, we have also neurons that are in culture not dividing, that is in the same pipeline. So we're not far away to be able to answer that. But
okay, so you mentioned, so that next is my question. So you mentioned about the transcription dependency. So that's I saw the inducible promoter on the GFP construct. So what is the implication that why is it transcription dependent? Is this a transcription coupled repair involvement, or what? Where the case is working on?
So we tried a couple of things there. So we know that the contractions are not affected by a knockdown of XPA. So if it was a transcription coupled repair mechanism, you would expect XPA to be required. You know, there's always the caveat that this was an siRNA experiment, and you know there might still be enough residual XPA there to be, but you know this is what the data is. Then, so what I think is rather more probable is simply that Cas9 tends to be inhibited by the presence of nucleosomes, and repeats are very, very good at positioning nucleosomes. And so what transcription would do in this scenario would be to break down the nucleosome structure, allow Cas9 access to the repeat, and then that would be the reason why it's transcription dependent. But that's me speculating.
Very good. So Nan, you've been quiet. You must have some burning question or something.
Not at the moment.
Not at the moment. Okay. If you say so, I think you're asking for your turn. And then if we have more time, then we can discuss further. Because there is a common theme, you know, between those two speakers that we have here today. All right. So Nan, are you ready to present your case?
Yes.
Okay.
Good job, Vince.
Thank you.
Okay, see my slide okay?
Yes.
Okay. Thank you for the invitation. I'm going to talk about two RNA cleaving modalities today. They are CRISPR-Cas13 and DNAzymes. Okay. So my talk is consisted of two parts. So one is on the Cas13 approach. I'm going to highlight some results on DM1. For the second part, I'm going to talk about some data on polyQ diseases. This is not the theme of the conference, but I'm hoping to highlight the application for the technology to be used in microsatellite expansion diseases. So we all know of myotonic dystrophy. It's a multisystemic disease affecting multiple organs. That's why it's been a problem for delivery for gene editing as well as oligonucleotide therapies. So the disease is classified as a RNA gain of function. So it's a missplicing and downstream processing. The goal currently is to eliminate the expanded RNA. There are multiple wonderful technologies out there through collaborative efforts, such as ASO, CRISPR-Cas9, such as Dion just talked about. You could use guide RNA cutting flanking with single guide RNA or double guide RNA. So there are dCas9 such as transcription blockage by Erica Wang's lab, and also dCas9 conjugated with pain by Dr. Jing Yu's lab. There are also small molecules as well So they tend to strip the regulators away from the expanded RNA, and also small molecules conjugated with RNA cleavage modules. So our strategy is slightly different. So we're trying to use CRISPR-Cas13 to cut the RNA. So Cas13 is an RNA-guided RNase. By convention, is classified into 4 classes from A to D. So the D class seems to have the smallest size, so preferably for packaging. The one we use is actually called Cas13 or C2C2. So the protein is quite large. It's close to 1400 amino acid. So it has 2 functional domains. The N-terminal domain is responsible for the guide RNA maturation, whereas the C-terminal domain is for target cleavage. There are 2 motifs responsible for the cleavage. They are HAP-N1 and N2. So if we mutate the HAP-N2 R1278 molecule into alanine, this renders the entire protein active. So this becomes a Cas13. So the 2 hapens are located close to the surface of the protein from the crystal structure. So they sort of form this loading channel, partially open. Some have suggested that this could be accounting for off-target cleavage. So the guide RNA has a 5' handle, which is a hairpin, and it's followed by a 28 nucleotide CRISPR spacer. So this CRISPR spacer can basically target any sequence. The only requirement is that the 3' PFS is not G. There are several advantages of using Cas13A to target DM1 CUG repeats. So first of all, we're not manipulating at the DNA level. So perhaps it's safer without permanent alteration. And the Cas13 preferentially cuts as uracils. So we know we have a lot of use in CUG repeat expansions. And because it's a protein, we can conjugate it to a specific promoter to achieve tissue specificity. And also Cas13 is compatible with multiplexing. So all you need is 1 promoter, it's a Pol3 promoter, to transcribe a polycistronic guide RNA. This is quite different from Cas9. For that, you need for every guide RNA, you need a single promoter. So with that said, Cas13 is not perfect. So it's got its own problem. For example, Cas13 has this called collateral damage RNA activity. So once it's activated by the on-target guide RNA, it tends to cleave any RNA in proximity. So this is something we bear in mind when we design the experiments, and also we want to assess the effects in our studies. So when we designed the experiment, we wanted to know which part of the DMPK to target within the RNA. So we could target the flanking unique sequences, but based on some studies, we know that CUG repeat itself could form gel-like foci and still trap MBNL. So that's why we decided to target CUG repeats directly. This is also encouraged by some ASO studies as well. But the problem come with it is that we could generate off-target effects because there are a lot of mRNAs that contain short CUG repeats in the genome. So the first objective of our study is to see if we can use the Cas13 to track CUG foci. So we cloned the cassette, this is the Cas13 cassette, with the EGFP conjugated to it, flanked by 2 nuclear localization signals. So we cloned this into a PX330 backbone. And within the cells, we can see that without a guide RNA, the protein localizes predominantly in the nucleus. And to target CUG repeat in DM1, we designed a guide RNA that's finishing with a 3' PFS with a uracil. So we put everything into this all-in-one plasmid, and we co-transfected with CUG 9600. This is a plasmid derived by Dr. Thomas Cooper. So it's an interrupted repeat. Every 20 CUG is interrupted by a cloning site. And we also designed a guide RNA that's non-targeting. So when we co-transfect the 2 factors, we can see that the Cas9 is actually tracking the CUG foci in the cells significantly more than the non-targeting. And to see if the Cas13 would track other components, other RNA-rich organelles, we focus on stress granules, because stress granules contains a lot of RNA as well as proteins. So we designed a guide RNA targeting ACTB mRNA. So ACTB mRNA and the G3BP1 are very enriched in stress granules. So with this vector, we transfect it into the cells and induce stress, and we can see that the Cas13 is tracking the stress granules. This is kind of interesting to us because the Cas13 has two analyzed signals, but what looks like is that the zip code of the guide RNA is actually pulling the Cas9 to the right compartment. This has been corroborated by other studies as well. The second objective of our study is to see if we can use the functional Cas13 to degrade CUG repeats. So we started with some biochemical cleavage assays. So we used purified Cas13 and in vitro transcribed CUG RNA. And we did an overtime over 16 hours. The cleavage efficiency is a bit slow, but we can see a clear difference between Cas13 itself and Cas13 with guide RNA. It's kind of interesting to us that the Cas13 itself can cleave some of the guide RNA, some of the CUG RNA as well. To see if the Cas13 could work in myoblasts, we designed an all-in-one vector. This is a lentivector. So the Cas13 is under the control of EGF1 alpha with two analyzed signals and puromycin resistant. This is on a lenti CRISPR V2 backbone. And as you can see on the right, we transduced the DM1 myoblast. This is a cell line provided by Dr. Gombi. And so in his recent study, it has been shown that this carries more than 1900 CUG repeats. And we can see there's a clear knockdown of CUG foci, as well as the size of the CUG foci. With these generated myoblasts, we wanted to know if we can reverse any missplicing effects. So we picked 6 RNA targets. These have been used as targets in Dr. Brown-Batra's paper. So with the DM1 targeting guide RNA, we can see that the missplicing is actually reversed in all 6 tested mRNA. And it's interesting that we see partial rescue with DM1 non-targeting. So this is quite consistent with our previous slide that non-targeting is actually partially reducing the foci number. And on the right, you can see that with Cas13, we wanted to know if it affects the DMPK mRNA level. So we did a qPCR in these cell lines. You can see the knockdown of DMPK is quite minimal. So this would suggest that our Cas13 is targeting the accumulated foci rather than the DMPK parental mRNA. We're also interested in the off-target effects. So we picked 2 mRNAs. These 2 mRNAs have been tested in Dr. Matthew Disney's paper. So they carry roughly 11 to 16 CUG repeats. We can see that we observe off-target effects with Cas13. So to conclude, we can see Cas13 is able to track RNA-rich organelles in both nucleus and the cytoplasm. We show that Cas13 degrades CUG repeats biochemically and in the myoblast. And Cas13 rescues missplicing in the treated myoblast. And we observe off-target at 2 levels. One is with guide RNA, non-targeting guide RNA. We think that this is quite different from the collateral, because in the collateral cleavage, you need activation, on-target activation. But with non-targeting, you don't have the on-target activation. So this must be coming from the Lash Cas13 itself. And on the second level, we see off-target on the CUG-containing mRNA. So for future directions, we want to improve the cleavage efficiency as well as precision with the new generation Cas13 orthologs. So these include like the recently discovered Cas13B, BT, or X and Y, etc. So these can be used for packaging into a single AAV. And also there's a new version of Cas13. Oh, sorry, not Cas13, Cas711. So this one doesn't actually have collateral damage. So we could use that as well. And we also wanted to assess if the cleavage can be achieved in multiple cell lines, as well as in vivo. And also we want to test if there are a preference for large repeat cleavage or intermediate or small. So that would require different cell lines as well. We also wanted to modulate Cas13 activity. So we have managed to clone an aptamer into this guide RNA. We're currently testing its on-off switch. So this leads to the second part of my talk. I'm going to talk about the DNAzyme I'm working on at the moment. So DNAzymes are single-stranded DNA. So they don't exist in nature. They are artificially evolved to have multiple functions. The one we're interested in is called RNA cleaving DNAzyme. So based on the crystal structure, you can see that the DNAzyme has two RNA binding arms, as highlighted in green. So we can easily swap out these RNA binding arms for different sequence specificity. And it also has a catalytic core. So this catalytic core is folding into this pseudoknot. So the folding is dependent on the cation presence. The biggest advantage of using DNAzyme over other oligonucleotides is that it doesn't require any protein components to assist the cleavage. So it autonomously cleaving its RNA target. So this distinguishes from ASO based on RNase H or siRNA based on RISC complex. So there are two predominant DNAzymes present, have been artificially evolved. One is called the 817 DNAzyme. The other is called 1023 DNAzyme. So as you can see, 817 specifically cuts between A and G, whereas 1023, which is supposed to be a derivative of 817, it cuts between R and Y. So R represents, that gives the R and Y cleavage would give a broader targetability of 1023. So it can be used for a variety of microsatellite repeats, such as DM1. So we could cut between two CUG boundaries with 1023. So DNAzymes can be highly reprogrammable. So that means they can be used for repeat sequences as well as unique sequences. And also is compatible with the packaging. So far, we have managed to package DNAzymes in liposomes, as well as conjugating to DNA origami. Another advantage of using DNAzyme is that we can potentially identify SNPs. So with these SNPs, we can cut with DNAzyme on the expanded allele over the non-expanded allele. So this kind of SNPs might not be applicable for other oligonucleotide therapies because the hybridization restraint would not allow this distinction. So our and others have identified that DNAzymes, so I'm talking about the A17 DNAzyme. So it cleaves the RNA in the presence of different divalent cations, such as magnesium and calcium. A lot of the cations are present in the cells. So we think that DNAzymes could work under multiple divalent cations to get a combinatorial effect in cells. We and others have also found out that the length of the RNA binding arms affects the catalytic activity. So in general, the longer the arm, the more likely you would have a tighter binding. And also the longer the arm, the more likely you would have a mismatch tolerance. So there's a trade-off. And we used a few modifications to improve the DNAzyme binding affinity as well as stability. So we used the LNA and the PNA. As you can see in this graph, we have comparison for in vitro cleavage of target with unmodified DNAzyme, LNA modified DNAzyme, PNA DNAzyme. So you can see that LNA DNAzyme seem to retain some catalytic activity, whereas the PNA DNAzyme does not. So this probably could be attributed to the peptide backbone that's not folding very effectively. So to see if the DNAzymes would work in cell lines, we transfected DNAzyme with HTTQ74 into HEK cells. So this is not unmodified DNAzyme. This is LNA modified DNAzyme, and this is PNA modified DNAzyme. So you can see at a protein level, the LNA DNAzyme works very efficiently. It's clearing about 80% of the target protein. And this is well reflected at an mRNA level. So in qPCR, we see a very similar pattern. So this would suggest that the cleavage is actually based on RNA cleavage rather than protein inhibition, as seen with some LNA ASO studies. And to see if the LNA DNAzyme could cleave other CEG repeats, we cloned several polyQ expansion proteins, and the efficiency was ranging from 70% to 100%. To see if the DNAzyme could work in neuron-like or neuron cells, we co-transfected HTTQ74 with LNA modified DNAzyme into neuroblastoma cells, SHSY5Y. We also did it in excitable cortical neurons. So these eye neurons derived from iPS cells. So we can see roughly 90% of target cleavage in these cell lines. And we also tried to see if DNAzyme would work in patient-derived cells. So we got SCAR1 patient-derived fibroblasts. We can achieve allele-specific knockdown of mutant ataxin-1. But this is not the case for SCAR3 patient-derived cells. We see roughly equal proportion of knockdown with mutant and wild-type ataxin-3 in SCAR3 iPS cells and fibroblasts. And finally, to see if the DNAzyme would work in mouse brains, we injected roughly 25 micrograms of Cy5 labeled DNAzyme into a SCAR3 mouse brain. So this is via ICV. So you can see that the DNAzyme seems to persist over a month, at least 42 days. And we increased the dose to 400 micrograms and did the intraventricular injection into the right ventricle of SCAR3 mouse. And in this figure, you can see that we can clear out roughly 50% of the high molecular weight ataxin-3. This is the mutant aggregates. To conclude, we know that DNAzyme works under different cations and in a number of cells. So it seems that it works very efficiently in neurons or neuron-like cells, and its activity decreases as we approach fibroblasts or iPS cells, but we can still get 40 to 60% knockdown of targets. And we did RNA-seq data. So we transfected DNAzyme into a wild-type fibroblast, and we didn't see any off-target effects over 60 mRNAs containing short CEG repeats. And we can see DNAzyme can achieve allele-specific knockdown in certain cells, and we don't actually see abnormal toxicological abnormalities or immune activations in the mouse treated. In the brain, we tested cortex and cerebellum. So for future directions, we want to optimize the catalytic domain and binding arms to improve the multiple RNA turnover. So that would increase our efficacy. We also want to increase the dosing regimen to improve the in vivo efficiency. And also, as I mentioned before, we managed to package the DNAzyme with the liposome, and the knockdown effect was a bit low. So we're trying to optimize the liposome with ionizable lipids so that it can efficiently escape the endosome. So I would like to thank a lot of people, so especially my division head, Dr. Ashizawa. He's constantly supporting my studies. And I also received a lot of cell lines from Dr. Denis Ferling, Dr. Guanbin Xia, Dr. Thomas Cooper, and Dr. Partha Sakar. And last but least, I would like to thank my funding bodies. Thank you.
All right, thank you, Nan. Can you hear me?
Yes.
Okay, very good. So I forgot to introduce Dr. Zhang. Dr. Zhang is an assistant professor of neurology at Houston Methodist Research Institute and the Weill Cornell Medical College. So Houston Methodist is a part of the Weill Cornell affiliation. His key focus is identifying therapeutic innovations and pathogenic mechanisms in neurodegenerative diseases, including myotonic dystrophy and other repeat expansion diseases. So he's developing two reprogrammable RNA targeting tools you heard CRISPR-Cas13 and DNAzyme in this presentation. And he's working on both in vitro and in vivo models and treating myotonic dystrophy type 1 polyglutamine disease. And also more lately, we are targeting SARS-CoV-2 virus. And so those do not have repeats, but we can meaningfully target viral genome, RNA genome. His previous research at the Imperial College of London, UK, involved structural elucidation of transcription complexes, protein engineering with artificial amino acids, and RNA damage repair. And he's very talented in a number of different technologies, particularly in vitro. And I think he's quickly learning about in vivo, a number of different issues. And I think here we are open to questions. Let's see. So Dr. Kumabehara asks, CUG expansion repeats are bound by multiple RNA binding proteins. How accurate will be your target? How much off target in CRISPR?
That's a very good question. So we think probably based on several studies, either it's Cas13, Cas9, Cas9, Cas13, it seems that the complex of Cas is able to kick off anything that's bound to the target RNA and then induce the cleavage. So when it comes to off targets, we're still investigating how to avoid cutting other short CUG-containing RNAs. So this could be coming from the guide RNA binding to these short repeats, or coming from the collateral RNA binding, collateral RNA cleavage. So it's difficult to assess how the guide RNA would distinguish the short CUG from the longer CUG repeats. It's possible that the longer CUG repeats would have more binding sites. So it would mop up the Cas13 or Cas9, and also the binding conformation might be favorable for these bindings.
Okay. So, I think that Nikkei's and your two approaches, and both targets a repeat. And in myotonic dystrophy, particularly is a repeat number differential between the disease state and normal state are very large. And that CAG repeat expansion, depending on the disease, the differential is not so great. And but in general, I think this is very interesting that repeat as a target may be viable in many situations. So I am wondering to then the disease is like a much smaller repeat expansion, and let's say SCS6 or some other diseases repeat is very small. Do you think this is still workable? And same question to Vincent.
So Nan, you want to go first, or?
Let me think about it. Do you want to go first?
I mean, if I can jump in first, then it is. So I think there's one thing not to forget with the other diseases as well is the somatic expansion, right? So like Huntington's is a good example of that, where you inherit a relatively short repeat track, but in the affected tissues over time, you end up with something that's quite large, not quite myotonic dystrophy size, but very close. And so it might very well be effective on the somatically expanded alleles that seem to be driving some of the pathogenesis anyway. And so
do you mean that there may be some advantage in targeting the more diseased tissue because they tend to have larger expansion?
To have larger expansion so that you can differentiate them better is what you mean?
No. I'm talking about you want to, for example, if you give Nikkei systemically and it may go deliver to all kinds of tissues. But that may actually selectively target the tissues that has larger CAG repeat expansion in Huntington disease.
Yeah, I mean, if you do it, well, systematic. So I would be more worried if you, because obviously the more cells you target, the better it would be. But then the more cells you target, the more off targets you're likely to induce as well, right? So there's a balance there. This is probably what I would be more concerned with. As to, I mean, and Darren has talked about this yesterday as well. The affected tissues in myotonic dystrophy are also expanding quite a bit somatically, and so I think we would be in a similar situation there, where there's still a lot of somatic expansions that occur in affected tissues in DM1 and presumably DM2.
Okay. So another question from Lynn Schneider. Oh, now go ahead.
Back to your question about SCAR3. Okay. So I think it's actually based on a lot of diseases. The targeting will be very differential. So for SCAR3, you could afford to target both wild type and mutant allele without affecting the genotype and the survivability. With SCAR1, this is not the case because I think based on Dr. Huda-Zogby's data, the mutant, if you knock down wild type, the mice would have cognitive defects. This probably would be true for Huntington as well. For Ataxin-2, if you knock down the wild type, it's actually beneficial in mouse models. So I think you have to depend on different disease models.
So Lynn Schneider asked, would this also be applicable for those of us with DM2 and?
Yes, it would be. Yeah. Okay. So we can definitely redesign the DNAzyme or Cas13 to target DM2 CCUG.
So already this question was asked to Vincent, but what do you think about the liver system? You showed some in vivo data, but what do you think there's a limitation? If there's a limitation, how do you overcome? Namely, do we need to use AAV type of thing, or just you need what I call locked nucleic acid mode, just directly inject into the tissue or ventricle, or what is your vision for the treatment? And in case of myotonic dystrophy, it's much more complex because it's muscle, heart, and that kind of things. And so, do you have any idea where you want to take it?
Can I jump in on this question?
Sure, no, yeah.
Okay, so I think the problem with the DM1, for example, AAV, you probably need a large dosage to achieve bioavailability in different organs. So this seems to be the problem because in recent clinical trials, you can see that with Astella or BioMarin, they actually have a problem with high dosage AAV. So if you inject like 2 times 10 to the power 14, you would have a lot of liver damage. So this has been shown like AAV, non-modified AAV accumulate in the liver. It would cause liver failure and sepsis, etc. And also recent study shows that in mouse models, if you use a high dosage, you would actually encourage AAV genome integration. So this has been shown with the BioMarin study. And also this is going to cause a lot of cancer problems. So this is kind of very difficult. As Vincent just said, there's a new Cell paper with a MyoAAV modification with the capsid. So this would actually encourage the widespread in the muscles and also reduce the liver spread. So this might be a future potential for us to employ.
So DNAzyme is very similar to antisense oligo, very structurally very simple and small. And so I was listening to Ability and Dion, in the pharmaceutical session, and their scheme of conjugation with antibody as well as IFAB, I mean, that's easily done in DNAzyme. And what do you think about those approach?
Yes, so I think the delivery for DNAzyme, if we were to use it in DM1, would again be the issue for reaching the right bioavailability So this has been shown for SO and stuff like that. So we have to find a way to package the DNAzyme to make sure it's released into the right muscles, into the right organs, tissues, and get released over time. So there seems to be other ways to get around this. For example, you could, in theory, package DNAzyme or ASO in that matter into AAV by relying on like certain vectors using reverse transcriptase. So these kind of systems have been published. I think we can probably employ these systems.
Okay. So I don't want to just dominate the questions, but do you have a question to Nan, or Nan, do you have a question to Vincent, each other?
No, at the moment.
I think the questions have covered what I had. Delivery kind of questions would have been good, but
okay.
I don't have anything specific.
Okay, so just I want to ask now one more thing, and that's been bugging me in back of my mind, is you mentioned several advantages of DNAzyme over ASO. In real in vivo clinical studies, I mean preclinical studies in mouse, so do you think those advantages are truly advantageous in the cell environment in vivo?
You're talking about comparison between DNAzyme and ASO?
And ASO, yeah, right.
Okay, so that's very tricky because we would have to assess the contribution of RNase H activity towards DNAzyme, because with DNAzyme you have partial hybridization between DNA and RNA. This could potentially induce RNase H activity, but in our design in particular, we have LNA in the flanking sequences, but we only have a small portion of DNA, unprotected DNA, and we have a huge loop of mismatch. So based on the crystal modeling, this seems to interfere with the cis-al phosphate in the RNA, and also this seems to interfere with the RNase H. So we think that maybe this is not the case for RNase H contributing to DNAzyme in vivo. Another limiting factor for DNAzyme is that you need a high concentration of divalent cations. So historical studies suggest that these divalent cations concentration is actually low in bodies. But we think that because for example, magnesium calcium influxes, these DNAzyme could function as a sink for these divalent cations. So they could potentially carry out the activity very efficiently.
Okay, so there was a comment from Batra, and he said Zolgensma is a commercially available, has a commercially available AAV9 type. That programs for AAV integration is only seen by biomarine in neonatal mice, and this doesn't seem to be true for long-term treatments for hemophilia or SMA in large animals or humans. Do you have any comments, Vincent?
Oh, sorry.
Or either way.
Well, so if I understand correctly, he's asking about the integration of AAV into the genome.
Yes.
Yeah, I mean, yes. So this seems to be an emerging issue, right? So obviously, if you're using AAVs, it looks like it might integrate more than we would have liked, and that was previously advertised. So that's going to be an issue long term, because if you start having things integrating everywhere, obviously, then you end up it's off-target mutations is what it is really.
Okay. Well, time is up. So I think Ron Butler has more questions, but you can probably chat in the chat room somewhere. So thank you very much for a wonderful talk. And I would like to thank the audience as well with good questions and keenly and tuned into the discussion. Thank you. So we conclude the session here.
Thank you.